Seguidamente recibimos al Dr. David Joseph, investigador de McGill University, cuyo trabajo ha sido clave en el entendimiento de los mecanismos neurobiológicos relacionados con enfermedades degenerativas. Su ponencia nos invita a comprender cómo el último hallazgo de neurociencia puede abrir caminos hacia tratamientos más efectivos y personalizados.
Thank you very much for being here. Thank you for inviting me as a guest speaker to Asunción University, to Asunción University's event. Here I will be presenting my two single author peer-reviewed studies based on my neurological research. So my first study came out in the prestigious top high-impact journal, the International Journal of Molecular Sciences, in the field of biochemistry research in medicine. The paper has had so far a very high impact. I received here, as you can see in the Associated Press, an award from the director of the Faculty of Medicine, Dr. Greg Semenza at Johns Hopkins. He won the Nobel Prize in Medicine in 2019 for the discovery of the hypoxia-induced factor. This was an evaluation of my work by a Nobel Prize winner in medicine, Dr. Harvey Alter, who discovered the cure for hepatitis C. He qualified me as a candidate for the Nobel Prize in Medicine based on my work. My work has been downloaded. It's called views here, but they're technically downloads, roughly 8,000 times in 5 months, which is a world record for a single author study. And first, most important acknowledgement, I'd like to acknowledge my mother, Dr. Megan Libra. She was a PhD in law, one of the top lawyers in Canada. She sadly passed away from cancer 1 year ago exactly, at the age of 50. I wouldn't be who I am today if it wasn't for her. She's the best person I've ever met in my life. So I will go through the basics for basic fundamental research in medicine. For you, since you are medical students, I will gloss over the work at a faster pace than for a general audience. We will be looking at translational control in protein synthesis initiation in disease. First off, as most of you surely know, DNA is the fundamental code of the human body, containing the genetic information necessary for the development of the cells of all living organisms. This is the flow sheet we will be going through for me to introduce translational control to your field. DNA is followed by messenger RNA, which contains the genetic information from DNA, copied, transcribed into messenger RNA, or mRNA for short. mRNA uses nucleotide molecules to carry the genetic information copied from DNA. Very basic so far. The mRNA contains a 5' mRNA cap, which protects the mRNA from degradation. mRNAs have a 5' cap structure consisting of a 7-methylguanosine, a nucleotide molecule which contains no genetic information. However, it is essential for the mRNA's function. A nucleotide molecule, as you can see here, containing the genetic information, the first in the mRNA sequence of its kind. You have the methylguanosine here. You have the first nucleotide with genetic information here. And following that, you have a triphosphate bridge that links everything together. A triphosphate bridge. Subsequently, we will look at the eukaryotic initiation factor 4E. The mRNA 5' cap structure is connected by intermolecular forces to the eukaryotic initiation factor 4E. Discovered by Dr. Witold Filipowicz in 1976. He is the founder of the field of translational control research, and he conducted his discovery in the Roche Institute in the United States. You can see his peer-reviewed paper at the Proceedings of the National Academy of Sciences right here in Aaron Shatkin's lab. Now, as we go ahead, we look at ribosomes. Ribosomes are macromolecular machines that copy genetic information from mRNA. Ribosomes use this genetic information to produce proteins, a process known as translation. Now, proteins themselves produce the chemical reactions which allow us to live. They are known as enzymes, hormones, and antibodies. They are responsible for all the functions and structures of the cell and the living organism. Proteins produce the chemical reactions which allow us to live and would otherwise be impossible without them. You can't convert oxygen into energy without proteins. Otherwise, we could not have energy to stay alive. Now we get to my specialization, what I'm studying exactly. 4-EBP proteins. 4-EBP proteins, it's counterintuitive, stop protein production. This is a 4-EBP. They were discovered by Kay and Lin et al. In 1994. 4-EBP proteins attach to EIF4E, a process known as binding. This binding stops the recruitment of ribosomes by EIF4E, a process known as inhibition. As a result, proteins can no longer be produced. Therefore, you have two competing factors. You have EIF4E, which binds to the mRNA 5' cap structure and directs ribosome to this structure to produce proteins. You also have 4-EBPs, which bind to EIF4E and stop EIF4E from directing ribosomes to the mRNA 5' cap structure. As a consequence, proteins cannot be produced. In order to be healthy, a process known as homeostasis, you need a balance of these two competing factors to have proper functioning in a living organism. 4-EBP2, my favorite protein, is a member of the 4-EBP family found in the brain at much higher levels than any other 4-EBP protein. 4-EBP2 controls all brain activity because it is responsible for regulating the production of proteins in the brain. All chemical reactions in the brain are regulated in one way or another by 4-EBP2, which is implicated in all neurochemistry. This protein, this chemical reaction caused by 4-EBP2 is disrupted. 4-EBP2 is found in the cells of all mammals. However, 4-EBP2 undergoes a chemical reaction called deamidation only in the neurons of the nervous system, as discovered in the paper of Dr. Michael Bidinowski in 2007 and published in 2010. So this chemical reaction stops 4-EBP2 from binding to EIF4E and in this way makes possible the production of proteins. So you have 40BP2, which stops proteins from being produced. When 40BP2 undergoes deamidation, 40BP2 cannot stop protein production in the brain. 40BP2 is not destroyed or destabilized by this chemical reaction, as is usually the case, but it can no longer complete its functions. Which brings me to my research, my findings. I set to investigate why this chemical reaction happens in 40BP2, specifically in neurons. 40BP2 is found in all the cells of the human body. In the skin, in the liver, in the brain, but only in the brain does 40BP2 undergo deamidation. In neurons, nowhere else. My question was, why? For 16 years, despite extensive research by the experts in the field, there was no credible explanation as to why this reaction happens specifically in neurons and nowhere else in mammals. This was 16 years of research. This is very important because why is my research focus so important to the field of medicine? 40BP2 deamidation significantly alters protein production in the nervous system and maintains the balance of protein production that is required for having a healthy nervous system. Any imbalance in protein production in the nervous system causes neurodegenerative diseases. Discovering what causes 40BP2 deamidation will allow us to control this balance of protein production and develop effective treatments against neurodegenerative diseases, including memory loss. A discovery that paved the way for developing effective treatments against neurodegeneration, as well as memory loss. My work. I started my research in medicine at McGill University, one of the top research universities in the world, in partnership with my company where I am a director in biochemistry, FLOGEN Technologies Inc. On August 31st, 2023, I achieved a breakthrough on September 13, 2023, when I was only 23 years old. Now, I saw that 40BP2 deamidation, specifically in the brain, is conserved. You see 40BP2 deamidation in the brain in humans, in monkeys, in mice, in every single mammalian species. And as you know, when something is conserved in your genes, in all species, that means it's very important for survival. Why only deamidation of this protein in neurons? My hypothesis was 40BP2 deamidation happens only in neurons because of the axons and their unique properties. Now, not all of you are neurologists, so you're telling yourself, what are axons? Well, axons are long extensions of the neurons which conduct electrical impulses that are crucial to nervous system activity. In order to discover and to experiment on this topic, I used immunoblotting, which some of you may know. Immunoblotting, otherwise known as Western blots. What does immunoblotting do? Immunoblotting separates proteins based on their molecular weight. There are 20,000 proteins in the body, and each of them has a specific weight. Western blots can detect my protein of interest, 40BP2, specifically thanks to antibodies which target the protein. You know antibodies? Antibodies which attack bacteria? Well, we developed antibodies in industry to target proteins of interest in studies. 4-EBP2 extracted from the nervous system appears as 3 bands on Western blots. So when the antibody attaches to 4-EBP2, you see a band on your page of results. This is what you get after immunoblotting in 4-EBP2. You get 3 bands. You get the band of complete deamidation, you get the partial deamidation in the middle, and you get the bottom band of no deamidation. You also need a loading control to make sure that the same amount of protein is used in all samples for consistency. I invented a method of calculation for protein deamidation. This method is called the deamidation ratio, which we renamed as the Joseph ratio, in my honor, yay. The deamidation ratio is the ratio between the complete deamidation band intensity, the intensity of this top band, divided by the intensity of this band. The band of complete deamidation is divided by the band of no deamidation in terms of intensity. Why? The intensity of the bands reflect the amount of protein you have in your sample. I used mice for my experiments. Why? Mice, in this case, were wild-type mice. Wild type means that the mice were non-mutated. They're mice you can find in nature, but we can serve them in the lab. Mice are always used as models. When you want to discover a fundamental mechanism in medicine, you use mice, not people, because you can undergo multiple experiments on them that you cannot do in regular patients. 1, and 2, their systems, such as their nervous systems, are representative of human nervous system. Mice of the same age and sex were used in order to avoid data variability due to age and sex differences. So I had wild-type mice, 2 months of age, and all male. Experiments on the central nervous system. I wanted to know if my theory that 4-EBP2 neuron-specific deamidation is caused by axons or not. So logically, I had to look at the central nervous system and I had to isolate axons from cell bodies of neurons from the rest of the neuron. Now, how do you do that? You are medical students, most of you. You look at the brain and it's a mess. It's neurons going everywhere, one amongst the other. How do you separate that? Well, what I did is I invented a new type of experiment. My first goal for my first experiment was to compare 4-EBP2 deamidation in the 2 parts of the neuron, the axons and the cell bodies. And what I did is I looked at the optic nerve and the retina. Why? This is not common knowledge, but the optic nerve is a large extension of the diencephalon, a division of the brain, and it is easily isolatable via dissection. The retina is also an extension of the brain, specifically the midbrain, the diencephalon, as I said. Therefore, the physical properties of the optic nerve and of your retina are identical to the neurons found in your brain. I isolated the axons in the optic nerve and the cell bodies in the retina in order to determine if 4-EBP2 deamidation is happening mainly in the axons. And this was the first time ever that this experiment was conducted. This experiment is my invention. So I invented the ratio first for calculations, and I invented this experiment for proving my theory. Now, the second goal of my first experiment was to compare the deamidation in the whole brain with the individually isolated axons in the optic nerves and the retina. So you have, in the optic nerve, you have pure axons. In the retina, you have pure cell bodies, the other part of the neuron. And in the brain, you have a mix of axons and neuron cell bodies. So I compared the rates of deamidation between axons, pure cell bodies, and the mixture of axons and cell bodies. And I wanted to see what I get. This comparison shed light on where exactly 4-BP2 deamidation is originating from. These are my experimental results. If you want to take pictures, this would be the right time. The results of the experiments are as follows. Deamidation is significantly higher in the optic nerve compared to its retinal ganglia. The star here means a significant difference in values. The optic nerve, the axons had much higher deamidation than the pure cell bodies. Okay? There was no significant difference between deamidation in the axons compared to the brain. There was a very significant difference in the rates of deamidation between the pure cell bodies in the retina compared to the brain. Therefore, the conclusion is deamidation in the central nervous system is coming from the axons. So, I confirmed my theory that axons are causing deamidation in the central nervous system. Now, I wanted to see if I can extrapolate this finding into the peripheral nervous system. My experimentation was as follows. I had to invent another experiment for the peripheral nervous system. The goal here was again to compare deamidation of 4-BP2 in axons versus cell bodies. The sciatic nerve and the dorsal root ganglia were chosen by me because the sciatic nerve contains the longest axons in the body. The dorsal root ganglia contains the cell bodies of the sciatic nerve. This nerve is far from the brain. So if my finding occurs both here and far away from there to here, you get a general idea that my finding is generalizable to the entire nerves of the body. I isolated the axons in the sciatic nerve and the cell bodies in the DRG in order to determine if 4-BP2 deamidation is happening mainly in the axons itself. And this was the first time ever that such an experiment was performed. So 2 original experiments in addition to an invention of a new algorithm in this study. Picture time. Results of the experiment in the peripheral nervous system. What is the finding here? 4-BP2 deamidation mainly occurs in the axons of the peripheral nervous system. The difference was even more massive here. 3 stars of significance. A significant value, a p-value of less than 0.001. If you know math, that's a lot. The 4-BP2 deamidation is significantly higher in the sciatic nerve where the axons are, and little to none in the soma, which are located in the dorsal root ganglia. Keynote, when I say soma, I mean neuron cell body. Those terms are interchangeable. The finding here corroborates the finding in the central nervous system. Both in the peripheral nervous system and the central nervous system, deamidation is coming from the axons. Now, I continued my study with a comparison of the central and peripheral nervous system. A comparison of the rates of deamidation was made between the whole brain where axons are mostly myelinated and the sciatic nerve where axons are mostly unmyelinated in order to determine the effect of myelination. What is myelination? Myelination is like a kind of sheath, a kind of layer of protection that is put on axons. And these layers occur mainly in the central nervous system. The peripheral nervous system is poor in myelination compared to the central nervous system. And since my study focused on axons, I had to look at the impact of myelination on this reaction. The conclusion was a very, very, very high value of significance. P-value less than 0.0001. Again, if you know math, this is a lot. The conclusion is that deamidated 4-EBP2 is more enriched in the myelinated axons of the whole brain compared to the mostly unmyelinated axons of the sciatic nerve. This led me to my theory. I have the data I need. Now I need to interpret it, the most important part of research. I have 3 conclusions based on my experimental results. Deamidation in the central nervous system is coming from the axons. Deamidation in the peripheral nervous system is coming from the axons. Deamidated 4-EBP2 is more enriched in the myelinated axons of the whole brain compared to the mostly unmyelinated axons of the sciatic nerve. I needed to study the science behind these experimental conclusions. Now, we do not need to go into the minutiae of the organic chemistry behind deamidation. This is long, and we are not chemists in this room, most of us. We are medical doctors. But what you need to know is that deamidation is, I conceived this organic chemistry mechanism. Deamidation is spontaneous. You do not need an enzyme to have deamidation in the body. It occurs in itself after a certain amount of time. This is what you need to get from this slide. Next. And it does not move. I think the screen froze. Thank you very much. 4-EBP2 deamidation occurs in asparagenes, 99 and 102. There are sequences of molecules inside proteins. The molecules are called amino acids, and they are sequenced numerically one after the other. There is one type of amino acid in the protein called asparagine. In deamidation, the asparagine becomes aspartate, which is another type of amino acid. The asparagines affected by deamidation in 4-EBP2 are asparagine 99 and asparagine 102. The deamidation half-life is, thank you, Jesse. All right, we're back. The deamidation half-life is the time required. Yeah?
Sorry.
You can put the music on as we wait. All right. So the deamidation half-life is the time required for 50% of asparagines to be converted into aspartates. Robinson et al. Is a genius. He's retired now. He was a biochemist who measured how long it takes for deamidation to happen in proteins. He looked at it in most proteins, but he never found deamidation in 4-EBP2. That was found later. And he found that deamidation has a half-life. Now, if you know your chemistry, half-life is the amount of time for usually an atom to go from, let's say, uranium to another type of atom. They calculate the amount of time it takes for 50% of uranium atoms to be converted into something else. Robinson used this concept for deamidation, and he found that deamidation has the same process of half-life as uranium, for instance. The proteins have also a half-life. Just like atoms, just like the asparagine molecule, the protein after a certain amount of time is degraded. And when 50% of proteins are degraded, that's when you get your protein half-life. So, you have deamidation that has a half-life. Asparagines degrade after a certain amount of time. You have proteins that degrade after a certain amount of time. It was experimentally found that 40pp2 starts to be detected 18 days after birth. Since deamidation is detected in 40pp2 18 days after birth, I postulated that 40pp2 deamidation half-life is equal to 18 days. I understood from all this that if you want to see 40pp2 in a cell, you need to have a protein that lives on average longer than 18 days. This is the key concept. Some proteins live for only 4 days and degrade on average. Some proteins live for years. If you have a protein that only lives 4 days, if you have a 40pp2 protein that only lives 4 days, you're not going to see deamidation because deamidation takes 18 days. But if you have a protein that lives for, let's say, 27 days, you're going to see deamidation because the protein lives long enough for the reaction to occur. Now, I thought, okay, you need 40pp2 to live long enough for deamidation to happen. 40pp2 deamidation only happens in neurons. Therefore, 40pp2 lives longer in neurons than in other cells for some reason. Now, me being a scientist, I immediately asked why. You can never stop asking why in science. This is the golden rule. So I looked at other proteins, and I found that superoxide dismutase, which you may know, superoxide dismutase is a protein that controls oxidative stress in the body, lives for only 4 days in kidney cells. But when a brilliant scientist called Borchelt looked at superoxide dismutase in neurons, he found that superoxide dismutase lived 1 year in neurons when it specifically underwent axonal transport into axons. Now, 40pp proteins, when they are found outside the neurons, have a half-life of only 20 hours. As determined by Japanese scientist Dr. Yamaguchi. Remember 18 days? 20 hours is not enough for an 18-day reaction. 40pp2 in neurons was found to have a significantly higher half-life in neurons versus kidney cells already, as determined by Kululia et al. So the Kululia in Dr. Kokas's lab in Greece found that 40pp2 does live longer in neurons, but he didn't know why. So based on my experimental conclusions 1 and 2, if you remember, that confirm that deamidation is specifically happening in axons, I determined that 40pp2 half-life is significantly increased in neurons because of the axons. And this was corroborated by the research of Dr. Borchelt. Axons increase the life of other types of proteins, and the same process is happening in 40pp2. This was my conclusion. Now again, great. 40pp2 lives longer in neurons because of axons, and this causes deamidation. Again, why? I keep asking myself that. Why? And I found I had to look at red blood cells, erythrocytes, to answer my question. Now you may think this guy is looking at nerve cells. Why does he go to red blood cells? Well, everything is connected in medicine. Proteins are degraded in cells by proteasomes, the UPS system. ubiquitin proteasome system discovered by Aaron Chehanover and Avram Hershko, two Israeli scientists in the 1970s. And proteins in proteasome-poor environments such as erythrocytes have significantly longer half-lives. So the less proteasome you have, the longer the protein lives. You see where I'm going with this? Axons are found to be proteasome-poor compared to the soma, due to limited ribosome biogenesis happening there. Proteasomal activity has been found to be higher in neurons without myelin. Why? When you have myelin, the current goes faster from one neuron to the other. When you do not have myelin, you have all across the axon, you have ion channels. You have much more ion channels. And these ion channels consume much more ATP, the currency of energy in the body. And higher ATP consumption in the cell results in higher protein production. When you have higher protein production, the cell will create more proteasomes to degrade these extra proteins. So neurons that lack myelin have more proteasomes than neurons that have myelin. Okay? So, remember my conclusion 3? There is more deamidation in neurons with myelin than neurons without myelin. Well, that means that there is more deamidation in neurons with less proteasome than neurons with more proteasome. Therefore, I concluded that the proteasome-poor environment in the axons is what makes 4-EBP2's half-life longer and deamidation thus possible. I also concluded that 4-EBP2 deamidation is maintained at different rates for neuron cell type. Now, this was great, and usually in medicine, people only look at peer-reviewed publications, and the PhD thesis of students is often overlooked if it's not published, and the master's even less. But there was this brilliant master student in the Faculty of Medicine at McGill, Richie Truong. He found data that actually corroborates my result. Remember, my theory said that deamidation is caused by axons? Well, Richie found that deamidation was also detected in muscle tissue. Richie assumed that this was because of the muscle cells. This was incorrect, because when you look at cardiac cells, there's no deamidation in cardiac muscle cells, and they're both muscles. They're both muscle tissue. But when you look at when you look at muscle tissue specifically, you find that there is an extremely high density of axons in muscle tissue. An extremely high density of axons in muscle tissue, ranging in the hundreds of thousands for 1 unit of muscle. So I saw that, and then I looked at cardiac muscle. Richie found deamidation in skeletal muscle, but found no deamidation in cardiac muscle. What is the difference between cardiac muscle and skeletal muscle? Well, in order for skeletal muscle to function, they need axons. Axons in neuromuscular junctions. The cardiac muscle does not need neuromuscular junctions. The cardiac muscle functions in itself, independently of innervation from neuromuscular junctions. So, you have no axons in cardiac muscle, and therefore no deamidation. And you have a lot of axons in skeletal muscle, and therefore deamidation. So Richie's detection of the deamidation in skeletal muscle, but not in cardiac muscle,
corroborated my theory. Everywhere you look, you find corroborations for this theory in the human body. Now, all this study brought me to Davis-Joseph's principle. It is, due to their proteasome-poor environment, axons increase the protein half-life, which becomes more significant than the deamidation half-life of asparagines, creating neuron-specific deamidation. The biological clock is now observed. Why do I say biological clock? Remember that deamidation is a timer of disruption. My contribution fundamentally changes our understanding of protein production by demonstrating that the axon is a regulator of protein production in itself. The axon causes deamidation in the 4-ABP2 protein, and by doing so, stops 4-ABP2 from inhibiting protein production in the nervous system. This is my contribution to the field of medicine. You remember this that I showed you in the beginning? This is what my experimental results and interpretation bring to the biomedical pathways of medical science. Axons create 4-ABP2 deamidation, which allows protein production to progress. Next steps. Why are my findings scientifically important? Because you guys may think, okay, great, 4-ABP2 deamidation is caused by the proteasome-poor proteasome-poor environment in the axons. But why is this of importance? 4-ABP2 is fundamentally linked to memory. 4-ABP2 is crucial to memory formation in the brain, as determined by Dr. Banco in 2005. Understanding the cause of 4-ABP2 deamidation leads us one step closer to understanding the inner machination of human consciousness, because pro-4-ABP2 deamidation is a crucial regulator of protein production in the brain. Proteins do everything in the brain, and 4-ABP2 controls if proteins are produced or not, as determined by Biochemistry in 2010. The absence of deamidated 4-ABP2 in the brain leads to the development of impairments in the brain, as found in Kokas et al.'s paper in 2013. Deamidation is related to aggregation and amyloid plaque formation, a process which is the root cause of memory loss in Alzheimer's. 4-ABP2 deamidation is indispensable to the survival of all mammals. As I said in the beginning, 4-ABP2 deamidation has been conserved in every single mammalian species for 90 million years. When Mother Nature keeps something for 90 million years in all animals, it is crucial for survival. Another reason why this is of importance is found in my second single-author peer-reviewed publication, which came out 3 days ago on April 27th in the same journal, the International Journal of Molecular Sciences, a very high-impact journal, very difficult to get published there. The unified theory of neurodegeneration pathogenesis based on axon deamidation. This is life. This is how all proteins are produced through this very, very complex flow sheet I created in this publication. And this information, my experimental results, and the critical analysis of 224 research papers allowed me to come up with this. So this is very, very, very interesting. I found, picture time. I found that 4-ABP2 deamidation is connected to Alzheimer's through multiple different pathways. You have primarily a problem in aging patients. Where does Alzheimer's, where does Parkinson's, where do brain-related diseases mainly occur? In aging patients. Based on my critical analysis that I went through, deamidation is overexpressed in aging patients. based on Shiluri et al.'s findings, which determined that aging reduces the activity of proteasomes. Proteasomes are already poor in axons. Proteasome activity goes down when you age. The less proteasomes you have, the more deamidation you have. More deamidation means less 40pp2 to control the production of tau proteins, amyloid precursor proteins, Lewy bodies, as well as dysfunctional mitochondria. Lack of 40pp2 leads to dysfunctional mitochondria based on the Kulolia paper. Dysfunctional mitochondria inhibits mitophagy, the recycling of old and obsolete mitochondria. Mitochondrial dysfunction causes oxidative stress. Oxidative stress impairs mitophagy even more. Oxidative stress contributes to Alzheimer's and Parkinson's. Mitophagy can stem this degradation, can protect the body from this degradation here, but mitophagy can no longer function due to mitochondrial dysfunction and oxidative stress. And this is caused by a lack of 40pp2 due to increased 40pp2 deamidation. Even more, Vidinosti et al. In 2010 determined that 40pp2 deamidation impairs synaptic transmission. Synaptic transmission activates NF-kappa B. NF-kappa B, which has been shown to protect neuron degradation in Alzheimer's. So excess 40pp2, as I found, creates a multitude of problems for neurons. This principle extends to the main diseases of neuron degradation. Now, why are my findings financially important? We have here, sorry, I'll go back a sec. We have a target here. I discovered a target to control the occurrence and progression of neuron degradation in Alzheimer's and Parkinson's. Forbes, in a very interesting article, determined that discovering a drug which can delay, not even cure, but delay Alzheimer's by 5 years can lead to $953 billion in profits. Now, also, this work will lead to subsequent publications. I discovered, based on this, a new and effective drug for effective treatments for memory loss based on deamidation. I'm synthesizing a drug based on my discoveries. I also invented a new method for studying the progression of disease based on deamidation and cancer. Remember when I'm talking about specific deamidation? 40pp2 deamidation is specific in the brain, but deamidation happens in 800 other proteins and hundreds of other proteins. And these proteins are all involved, many of them are involved in different types of cancer. Therefore, my research paves the way to new methods of study in cancer. I developed a new method of experimentation based on my research for studying cancer and deamidation together. These results are on their way for publication: deamidation in cancer and the effectiveness of my synthesized drug. The next steps, wow, I think I missed a slide. Wait a sec. Discovered. Yeah, we're here. I was going too fast. Okay, synthesizing the production of the deamidation drug I am in the process of creating is a crucial next step. We were in discussion of opening a lab in Paraguay and manufacturing in Paraguay, which I believe would be a very good contribution to this amazing country's economy. Such a drug would have a market value of roughly $70,000 per patient, $70,000 per patient, based on the data, roughly $70,000, based on the data provided by the previous Forbes article. And since, like I said, deamidation is a chemical reaction which occurs in other proteins, and these proteins are involved in cancer, my research paved the way for looking at deamidation's impact on cancer disease, which is a whole nother field. Drug production, therefore, will involve cancer prevention drugs as well. The drug I have synthesized for Alzheimer's control will be modified and reproduced for cancer subsequently. So, for me, that's it. If you have any questions, feel free to ask. Thank you very much.
Muchas gracias.